forward. There we go. Okay. Now we're better. Welcome everybody to the Centers of Excellence in Iron and Heme Disorders seminar series. Today we're privileged to have 3 core directors and staff to present to us some updates on what's happening at cores. The presentations will be monitored in the chat. And if you have questions, I want to invite you to populate the chat with those questions, and I'll monitor those as we go along. If you are not muted yet, besides the presenter and myself, I would ask you to go ahead and mute yourself. And our first presenter will be Hector Bergonia, who runs the heme analysis at the Iron and Heme Core at the University of Utah. We've been having some presentation problems, but for now, I will be presenting, and Hector's going to see those, tell me to advance the slides. So let me know if it looks okay.
Okay.
Is this screen okay, Hector?
Yes.
Okay, great.
Laurie, can you lower the volume? I have problems with muting. Okay. So hello, everyone. I'm here to, well, this presentation is essentially the same material, or very much similar material that has been presented to you by Laurie a few times before. I'm also here to touch on questions that have arisen about how we come up with the numbers that we send you. The Iron and Heme Core exists as a shared resource facility to help researchers working on heme and iron. Next slide, please.
There we go.
Okay. Now, Dr. Laurie Jackson and I make up the Heme and Iron Core team. Well, Laurie is Lady Iron. So why should you think we can even touch your super precious samples? Well, Laurie has extensive research experience. And on top of that, she was trained by Agilent on the ICP-MS. And she has been at this for about 6 years. I myself have been working on tetrapyrrole biochemistry with Dr. Phillips for over 2 decades. And I have been using related analytical biochemistry techniques before that, like HPLC. Next slide, please. Now, this, in a nutshell, is what we do. As heme, porphyrin, or metal analysis was performed at the Iron and Heme Core facility at the University of Utah, supported in part by a grant from the NIH, National Institute of Diabetes and Digestive and Kidney Diseases, grant number U54DK110858. This is how those who send us samples for analysis should reference us. Please put a version of this quote in your reports or papers that contain data that you obtained from us. Next slide, please. Oops. Anyway, this is, when I said heme, porphyrins, and metals, we also analyze those things in the process of measuring the activities of all the enzymes in the heme biosynthetic pathway. So we have delta aminolevulinic acid synthase, aminolevulinate dehydratase, PBGD or porphyrilinogen deaminase, uroporphyrinogen 3 synthase, uroporphyrin decarboxylase, coproporphyrinogen oxidase, protoporphyrin oxidase, and ferrochelatase. The uroporphyrinogen decarboxylase has several products, from heptacarboxyl porphyrinogen to coproporphyrinogen, and are all analyzed as requested. That is 7, 6, 5, and 4 carboxyl porphyrinogen. Now, the heme biosynthetic pathway evolved to make heme. Hence, for maximal efficiency, there should ideally be 0 steady-state concentrations of the intermediate. But since no process is ever 100% efficient, that's the second law of thermodynamics, we may expect very small amounts of intermediates in normal, unperturbed systems. Some people who sent us samples were questioning why the intermediates were so small, especially the heptacarboxyl porphyrinogen to pentacarboxyl porphyrinogen. Next slide, please. These are our major equipment. It consists of a Waters Acuity UPLC system and the Agilent 7900 ICP mass spectrometer system. The Waters UPLC system consists of a reverse phase C18 column with temperature controlled, a fluorescence detector, a photodiode array detector, a binary solvent manager, sample manager, and the samples could also be temperature controlled. These are for the heme biosynthetic pathway intermediates. The Agilent ICP-MS system is for metals, usually iron, but copper, zinc, and manganese are commonly also quantified, and many more elements of the periodic table are also analyzed. The advanced separation principles used in these two have actually been introduced in the most rudimentary form in your high school or college freshman years. For the UPLC, it's like dissolved light, it dissolves light. While in the Agilent, you have the Newton's second law of motion. For the UPLC, the more hydrophobic a molecule is, the more it is slowed down by the hydrophobic C18 coated particles or the stationary phase in the column, and hence are separated from the faster eluting, less hydrophobic molecules. The complications are the molecules can have both hydrophilic and hydrophobic parts, and the overall hydrophobicity often depends on pH when some moieties are ionized or neutral. Temperature, organic content on the mobile phase also affect separation. In the mass spec, for Newton's second law of motion, it just states that a heavier particle needs a greater force compared to a lighter particle for both to be deflected the same amount when passing through a force field, like an electric field in the mass spec. The variable force in the mass spec is applied to a metal ion stream such that only particles of a specific charge to mass ratio are allowed to reach the detector. The others are filtered or deflected off. The challenge here is that you really need the metal ions in the plasma stream to be discrete single atoms with no multi-atom particles with identical charge to mass ratio. Next slide, please. Now we have here the ancillary equipment. This is the list. Well, it's most of it. I'd like to call your attention to the Atmos bag, which is actually a plastic bag, essentially. It is a poor substitute for a true anaerobic chamber, but it is good enough to allow work in an inert atmosphere like argon to absent oxygen to, for example, chemically reduce porphyrins to porphyrinogens, and especially to assay for protoporphyrinogen oxidase. I also always have to do this under minimal illumination, since light can stimulate oxidation with any remaining little bit of oxygen in the system. Next slide, please. These are, needless to say, the substances requested for analysis are almost always not measured as they are directly. They are often converted to other compounds that are more conveniently measured. ALA, for example, is converted to a fluorescent derivative. Porphobilinogen is converted to either a highly colored derivative and measured for absorbance, or converted enzymatically to porphyrinogen and then oxidized. Porphyrinogens are oxidized to fluorescent porphyrin. Porphyrins are direct fluorescence detection. Heme, we never analyze for heme directly. It's usually in oxidized to hemine or iron 3 protoporphyrin 9 chloride. If this were hydroxide, that would be hematin, but we do hemine. Heme is also demethylated to form a fluorescent porphyrin mixture. And heme is converted to pyridine hemochromogen, where we look for heme A, B, or C. The ferrochelatase product is not, well, in the enzyme assay, it's not hemine. It is actually fluorescent zinc mesoporphyrin 9, where zinc is a lot more stable than iron during the assay. Okay. For the metals, well, they're actually a stream of positive, preferably +1 ions with a specific mass to charge ratio. Other compounds are also requested, like N-methyl porphyrin or biolins or flavins. Other substances that, other substances that can be analyzed by HPLC. Next slide, please. Here is another illustration of the heme biosynthetic pathway. You see, we do not analyze certain products of the heme biosynthetic pathway for practical reasons. We do not analyze the water produced, the acid, and the carbon dioxide. Note that in this system, if you carefully count, there are 14 carbon dioxides put out for every heme molecule produced. 1 for each ALA, and 8 of them going into uroporphyrinogen. Then 4 carbon dioxides from uroporphyrinogen to coproporphyrinogen, and 2 from coproporphyrinogen to protoporphyrinogen. Electrons for reducing equivalents are also produced in the oxidation of the porphyrinogen ring to the porphyrin ring. It is unknown if these reducing equivalents directly go to dissolve O2 or do something else in the cell. Next slide, please. So this is the link to the iron and heme website. This is a more detailed description of what we can do for you and how to make a work order, prepare or send samples. It is very important to communicate with us about your samples, as a few details of your situation may not be covered by the information in the website. Next slide, please. This is an overview of our website. Here we have our address where you send your samples. We moved here about a year ago, and the tabs on the left contain links to various sections: overview, facility description, referencing us, services, pricing, sample requirements and shipping, shared protocols, account setup, and contacts for iron and heme core. Next slide, please. This account setup tab gives instructions on how to set up a user account and to place an order. So placing an order puts you actually in the work queue. Right now, there are 4 orders on the heme queue, right? And I'm working on a huge set of samples that will take me a few more weeks to finish due to multiple tetrapyrrole and enzyme analysis requested for the samples. Next slide, please. Here, this sample requirements and shipping tab lists the types of samples that are currently sent to us and how they should be prepared. The cells, as to the cells, we do not really care about the count unless you want data in units per million cells. So we just want the 100 microliters approximately. The required amounts are listed as guides. The most important instruction here is send enough material. You see, perturbed samples tend to have higher amounts of pathway intermediates present and lower than usual enzyme activity. Next slide, please. The shared resources protocols tab contains links for general methods we use. These are often modified in minor ways to adapt to the samples we get. Sometimes the samples are a bit small, so I have to adjust volumes and things like that and still get good results. The principles behind the assays remain unchanged though. And the protocols are free to everyone to use. This tab lists the protocols on almost everything we do for those who work on iron and heme. Next slide, please. And here is a salute to them. Actually, I saw this bubbly listed in the Roots Diner menu. For our circle, it can refer to the iron biochemistry and biology worker. Outside, of course, it is the wife of the founding couple. Ferrarius actually means blacksmith. Thank you for your attention. Questions? Are there any?
Thanks, Hector. I have a question. Do you measure heme breakdown products, or can you?
Yes. Well, essentially, we can measure the bilins.
Okay. And
Yeah, we've done that before.
okay. Do you measure heme oxygenase activity?
No, not yet.
Okay.
But sometimes there are people challenge us on what we can do. Like, for example, somebody asked me whether I could do reverse ferrochelatase, so I did it.
Okay. Any other questions? I'm trying to get my cursor up here and stop sharing. Okay, thank you so much, Hector, for your presentation. Our next presentation is going to be from James Fox. I need to load this. Let me get it going. I'm sorry about this for everyone. Just a minute. Okay, no, all right. Hang on just a minute. My fault here. Let's see. James, for some reason I am not seeing your presentation. Sorry for this frustration, you guys. James, do you want to try and share your screen again? For some reason, my email is not, it's just opening up a forwarded announcement and not your presentation.
Try it, send it again, but share screen.
Oh, I got it.
Okay.
Maybe let me just get there. My fault. Okay. All right, can you see that?
No.
Oh, sorry. I'll mute mine.
Okay, let me just, sorry, I just had that one open. Okay, this should go. Now, let's see. I think it's this one. Let's try it. Okay. James, you're muted still.
All right. So thanks, Hector. Good morning. So today I'm going to give you a little bit of an update on the metabolomics core. We had some changes, not a lot. We would hope to have done more, but COVID was kind of an issue for our lab and for everyone else. But next slide. Interestingly enough, just COVID, we were busier than ever. It was a really weird year. Everyone had very odd years. And we actually had a 34% increase in samples that we ran. And the reason why this was we use auto samplers. And so what would happen was we just had people work independently in the lab at my core and set their samples on an auto sampler and run it. The auto sampler, they would go home and just monitor at home and process data at home. So actually, it wasn't terrible. So the past year, we had 43 University of Utah users compared to last year, which is not quite as many. A lot of CIHD users. We had 14 total, 7 internal CIHD users that were separate from the 43 university users. And we had a good amount of external users from the CIHD, and some of these are on the phone. So there's some Yale people here, IU people, and a couple other people use our core. And then we did some outside work for people all around the world. Actually, we try not to do a whole lot of outside work. And the reason why that is, we just don't have time. So this is the nice thing about the CIHD is it funds a person in the lab to be able to process samples from people outside the University of Utah. And actually, I think that's the bottom line I'm trying to show here. That's the power of this funding mechanism. And a lot of these were funded through pilot and feasibility projects, and some are ongoing still, some are done. But it's been a pretty interesting year to have as many samples. So that was an update on this. So let's just go through what the core is about. So next slide, please. So who we are here at the core, we're fairly small. We've actually shrank a little in the past year. People have moved on to either med school or more professional jobs. Our metabolomics people are Leon Catrell, who's been with me like 5 or 6 years. He handles all the LCMS metabolomics, and you probably, if you've done any work with us, you've had interactions with him. And Tyler Van Rye went to med school, Michigan State, and we replaced it with Quentin. A lot of people have worked with Tyler over the past few years, but Quentin's really capable. He has 8 years of GCMS experience, so he fills that hole pretty well. And so if you see emails, if send samples to us instead of Tyler, you'll be dealing with Quentin instead of Tyler. Our lipidomic side is a really popular side of work line that we have. We process hundreds to thousands of samples a month through lipidomics. And one guy really does most of that work. That's Alan Mashik. We're actually trying to hire another person to help Alan with all that work. But if you do lipidomics with us, you interact with Alan. And Kevin will talk a little bit about some of the proteomics we do, but we do have one person in the lab doing proteomics. That's Sandra Osborne. So we have a position open. If anyone here is at the U of U and knows a good technician looking for a position, it's an entry-level position. And if anyone knows a good data analyst who knows mass spec, send them my way, because we lost Brad Naylor, who some people worked with last year. But those guys are kind of unicorns. So if you can do computer programming and mass spec, then you're probably working at Pfizer or someplace. Next slide, please. So this is getting to the meat of what we do. It's the same we've done in the past 15 years. We continuously improve what we do. But the basics of what we do metabolomics and lipidomics, both in a targeted and non-targeted manner. And I'll discuss that in a second, using gas chromatography, mass spec, or liquid chromatography, mass spec for metabolomics. For lipidomics, we almost exclusively use liquid chromatography, mass spectrometry. It just works really well. So really to give a sense of the difference between non-targeted and targeted metabolomics uses what we call a triple quadrupole mass spectrometer. And the triple quadrupole mass spectrometer has a really high signal to noise level, but you pay for that signal to noise you have less metabolites measured at any one time. So you have to program into the instrument what metabolites you're interested in, and we then go analyze those. And if we have stable isotope internal standards, we can actually quantitate those really well. And we can do absolute quantitation if you wanted to go that route, which is more expensive, or just relative quantitation. And this kind of work is really appropriate for the majority of metabolomics we do. And the reason why it is most investigators really aren't doing biomarker discovery. They're looking at changes in redox levels, amino acids, really specific classes of metabolites. But the downside is we're only observing the metabolites that we're interested in. The upside is it's quick. It's cheaper because we don't have to spend as much time doing the data extraction and analysis. It's probably 10-fold faster in data extraction. But we also are funded to do like biomarker studies. And so biomarker studies, you really don't care what metabolic pathways are altered, or you just want to see what's globally changed in your mutant mouse or your cell culture or whatever. And we use non-targeted analysis. And with that, we use something called a quadrupole time of flight instrument. And we can see and observe 1,000 or more molecular features or putative metabolites, sometimes up to 2,000. It just depends what we're looking at. Most of those, or half of those, are lipids. But what we do with that is we're just agnostically searching for any alterations. And then once we find alterations, we go back and identify it. And that's great if you're doing cancer biomarker discovery. We have a project on now with like several thousand patient plasma samples. It's going to take 2 months to complete, but it's going to take us 2 or 4 months to take that data and make any sense out of it, right? So the downside of the non-targeted is it takes a lot longer of effort, both on the instrument and in our hands tracking the data. But like I said, horses for courses, they do different things and people have different desires. We do a lot of fluxomics and isotope tracing analysis, and this has become we usually have a project on at any one time on an instrument doing isotope tracing or flux. And the reason why this is useful is we are looking to see what metabolic changes occur over time in a system if you add a drug or a genetic mutation. And so fluxomics, and this is really kind of used interchangeably with isotope tracing, they're different. So just to be a little clarity, fluxomics is usually, it is a time course. So you feed in a C13 labeled precursor, usually glucose. In our case, 90% of our work is using C13 labeled glucose to a mouse, to a human, to a pig, to cell culture. And over time, we take time points, sampling time points, and look and see the use of glucose and the increase of downstream products, alanine, pyruvate, lactic acid, whatever else we're interested in the metabolic pathway. And we're fitting mathematical equations to see, what the rates between these pathways are. The difference between that and isotope tracing, we really don't care at all to do metabolic modeling. We don't really care about mathematical equations. We're just seeing if like we add a drug to a cell culture and have a control, what pathways are changed what pathways are changed from the addition of that drug, and not just in gross amounts, is there more glutamate or glutamine, but how much more carbon is being incorporated into glutamate or glutamine from the addition of labeled glucose. These don't require a lot of math. It's very easy. We're just looking at mutant versus wild type ratios. And most, if not all, I'd say 95% of what we're doing is actual isotope tracing, just because it's just as informative almost as fluxomics and way, way cheaper and way, way easier.
So James, this is Diane. I'm just wondering if you could mention an example that a CIHD member has used in some of this.
I do, actually. I'll show that at the end a little bit. But
Perfect.
we do have Harry Daly, Amy Medlock's group at University of Georgia. We've done a lot of the work with that. Isotope tracing, not metabolic flux, since we didn't fit any equations, but the paper in Blood in 2017, and there's another paper coming out in Blood Advances next week or 2 weeks from now that we did isotope tracing. So I'll show you that reference for that in a minute. We do metabolic. One more forward.
I didn't think I hit anything, sorry.
Yeah, so we also do metabolic quantitation. And so this is where we actually take metabolites. If you're interested in metabolite, one thing we're working on right now, it's been a bit slow due to COVID, is building an assay to quantitate 2-bisphosphoglyceric acid in red blood cells. And so what this is like an investigator just wants to look at a single metabolite and find the absolute quantitation of that metabolite, or peptide. We do peptides as well. And what we use there is we use a triple quad assay, and we use stable isotope labeled internal standards to build calibration curves. And so this is a really good way to get at absolute quantitation. It takes a lot of work to set up. Once it's set up, it's fairly easy to do. But the effort that goes into building a correct assay, it takes some time. Next slide, please. So I'm not going to go over all the instruments we have, I think we have 10 to 13. It just varies by the month. We haven't got a new instrument lately, but we were getting 2 in the next couple of months. Another GCMS for isotope tracing, and we'll get another QTOF. I'll show you in a second. But we have a number of instruments that do both the targeted analysis and the non-targeted analysis, depending on what you want it to do. So next slide, please. And so because we've been fairly successful, especially in the lipidomics arena, CIHD is going to place one of their latest and greatest instruments, or their latest and greatest instruments, into the core to hopefully build a couple more lipidomics methods for them. And we're actually going to use it for microbiome as well. And it's a Xenotop 7600. And what this will offer is greater increased sensitivity, which is always welcome. So everyone's got smaller and smaller sample amounts, unfortunately. And for us, what we're going to use a big chunk of this for in lipidomics is it has a different fragmentation method that will allow us to determine the position of double bonds in lipids. So that's always been a problem in lipidomics. We haven't been able really to get a full characterization of the lipidome because double bonds and acyl chains could be at a number of different positions, and we're unable to really determine those exact double bond positions. And so with the addition of this instrument, we will have a really complete lipidomics profile, if you like it or not, of your biological system, and tease out differences that can occur from the position of the double bonds within the acyl chains of the fatty acids. We're going to use it in microbiome research as well. And the reason we're going to use it in microbiome research is there's a lot of unknown metabolites in the microbiome. When we do microbiome work, which we do quite a bit of, not so much over the pandemic, but previously we've done microbiome research. Good 70% of the metabolites, we have no idea what they are. And so this orthogonal instrument, with orthogonal fragmentation pattern, will hopefully help us tease out a lot of that work. And so that's what CIHD is hoping we'll do. And so they're coming out August 16th, if you're here at the University of Utah, and having a demonstration of this instrument, and a speaker here as well. The speaker, she was a graduate student, Will Holland, here from the U. So it should be good. So that is, actually, I'm really excited about that additional technology. Next slide. So just to go back through how you submit samples, this is what's important to investigators. The most important thing is email me and let's discuss your samples, and I'll forward the email, or we'll talk on Zoom with the person who's probably going to do that analysis, either Leon or Alan or Quentin, because every project's different. We've done a lot of work with various biological sources, but not every single one. So we like to discuss, sample numbers, cell counts, amount of weight, what can be expected. If you have low cell counts or low cell numbers, we can get some metabolomics for you, but the more biological material you have, the more metabolites you'll get, which is a concentration issue. So once we've discussed all this, we have a sample submission portal, and it's the same one that Hector showed, chd.cores.utah.edu. You go there, instead of hitting the iron and heme core, you hit the metabolomics core, and there's a couple key things here on this webpage. One is there's sample submission form and submission details. And so it's important to download each one of these forms, completely fill them out. And when you go, next slide, to submit these on our sample portal, you will choose the lipidomics targeted or lipidomics non-targeted, either one you want. And it will ask you to upload those forms completely. And actually, we won't start the project until those forms are uploaded. And so we use those forms in the upload to populate the auto samplers or the instruments queue. So it's pretty important for us to have that. Right. So another thing about this, we're working on this right now. It's almost up and going. This is the improvements that we've been making in the past couple months. Is when you hit the submission for your project, you'll get an email with a 2D barcode in it. And what this is say you're out of the University of Utah, or even if you're at the University of Utah, we want you to print this out and put this into the sample box that you submit to us with your samples in it. And that way, when we receive it, we can open it up, hit that barcode with our barcode reader, and then it will populate where it's going to go into our -80 freezer. And then it will tell the software your samples have been received. And so then we're going to use that barcode throughout the whole process. And eventually we would like to have it set up within the year where the investigators will get an email, your samples are at this stage. Maybe people might be inundated with too many emails, I don't know. Or we'll have a link like this is where in the process your samples are at. So that's been a weak point of our sample submission process. It's been kind of a black box. We don't want to do that anymore. And so we are working at improving every step of the sample submission process. And that's kind of nice. With COVID, we were able to do that because people can program from home. So next slide. So this is some recent examples. The one I was talking about, that blood advances, the immunometabolite, it kind of inhibits heme synthesis. So that's a really great paper illustrating not just the mass spec metabolomics core, but also the iron and heme core. And this work was done with John Phillips here at the University of Utah and Amy Medlock and Harry Daly's lab at the University of Georgia. So it's just a really nice paper that encapsulates pretty much everything that these cores do. We also had some work with Carolyn Philpott at the NIH with iron work, looking at iron chaperones. My lab had a publication looking at how to improve throughput. Next slide, please. And so what we're doing now is, like I said before, absolute quantitation of the 2,3 BBG or DPG, just depends on who you're talking to, using custom made internal standards. So this is actually interesting. There used to be an assay, I think made by Roche, that they don't sell it anymore for this particular metabolite in red blood cells. And so we were approached by Tom Gans and Shilpa Sharma to build an assay for this. And so what we did, you couldn't buy the internal standards. So actually I had it custom made by Cambridge Isotope. We got that in-house, and right now we're building the assay for it. The other thing we really want to do, and we've actually put a lot of effort into this when we can, is we building our targeted assays. We actually have a number of them already. And so we've had numerous publications where we use targeted assays for people to quantitate their favorite metabolites. But what we're doing here is we're focusing more on the red blood cell to do triple quad quantitation of red blood cell metabolites. The reason why we want to do this, it's more sensitive. And once we have it going, it'll be much more rapid. It's built out right now. We can do this, and we do this for cell culture and some red blood cells, is we have 75 metabolites. We can have 65 metabolites in the positive mode that we can observe and 70 in the negative. We'd like that to get to about 100 each in the negative and positive, and then add as we can go, or add more as necessary. And the real advantage for the investigator with that is the turnaround time is going to be much faster, because the data analysis goes from like taking 10 hours to less than 1 hour. And all our report buildings, it's automated. The downside is you're not going to find anything new or novel metabolites, but you'll find metabolites you're interested in. And like I said, we're improving the sample tracking for investigators. And so with that, I'll take any questions.
Great. Thank you, James. Appreciate that. Any questions that people want to ask about in there?
James,
Yeah.
Do you deal with stereoisomerase?
We don't. So that's a real
Some
specialized
metabolites, they arise.
Yeah, so we have done like 2-hydroxybutyrate, we have an assay for that, both LCMS and GCMS. That's been a couple of publications, but for the most part, we don't really do it.
Okay.
And the reason why we don't have a lot of people asking for it, actually.
Okay, thank you.
Okay.
Hello? Hi.
Hi.
Can you hear me? Hi,
Yeah.
James. Yeah, so I'm working on T cells. So I'm interested in to analyzing this metabolites from the T cells samples here. So my question is, normally how many cells you need to do
So
this measurement?
That's a real good question. We find immune cells are difficult. They are not as metabolically active, I think. Some we just did it depends on what you want to do. So the more targeted the assay, if you tell me more like, we want to look at this set of metabolites, the less cells you need. So that's just the way these instruments work. If you want to measure 200 metabolites using a targeted quantitation, you need more cells. If you want to measure 20, you can use less cells. It's a sliding scale. You almost have to do a preliminary study. Macrophages, I know they're not T cells, but
yeah, T cells are smaller, much smaller.
yeah, but macrophages, for whatever reason, they're low metabolically active, I think, in some states. So we need at least 1 million. But it really depends. So we really have to think about what you want to observe.
Yeah.
Yeah.
Okay. Yeah, I think I will email
Yeah.
you and to discuss the details for our project.
Yeah, definitely. It's one of those things, like you do see people publish metabolomics on cells that, but they're pretty narrow on what they're detecting.
Yeah, once T cell activated, they will upregulate their metabolic programs.
Yeah.
So then we can detect more metabolites from this activated T cell.
Yeah.
Okay, thank you.
Yep. Yes, sir.
Other questions? Please feel free to chime in and ask if you have them. Okay, otherwise, thanks, James, very much. And we'll move on to our next speaker, which is Kevin Hicks, who is going to talk to us about the new, what we call Midas core at the University of Utah, CIHD. I'm just working, Kevin, to get that presentation showing. Let me get my screen shared. I think we're there.
Hey, Diane, if possible, is this the compressed? Did you get my email with the compressed version? It removes all the animations and everything.
Beats me. Let me just check that. Sorry.
Yep.
Let's see. I got an email. I got 2 different emails. How do I know if it was compressed?
It should be the most recent one. And the slide deck it'll be the same name, but it'll say compressed on the end. Otherwise, I'm going to be telling you to click next
A lot
hundreds of times. So
one of them is 18 megabytes and one is 9.
use the 9 megabyte.
Okay,
Please.
let me just get to that. Let's see. Can you see that?
I think we're still on the other, on the large 18 megabyte file.
Oh, okay. All right. Let me try again. Oh, thanks everybody for your patience in
You may
this.
need to stop. It looks like you're still sharing the first part. There we go.
Okay. All right. So let me go in again. I think. Let's see. I'm not sure why this struggled today, but it's all good. We'll get there. So hold on just 1 minute and I think we'll get there. Okay, so let me see if I can share my screen now. Okay, can you see that?
Yep.
Okay, so I'm going to go into the presentation mode. Is that
Great.
what- Okay.
Yeah, that looks perfect. Awesome. Thank you, Diane.
Yep.
Well, hi, everyone. I'd like to introduce myself. My name is Kevin Hicks. I was a postdoc in Jared Rutter's lab here at the University of Utah. And during my postdoc, I developed a novel platform for the non-biased discovery of interactions between a target protein or proteins and a defined library of human metabolites, which kind of represents the human metabolome. And this is the first presentation to the Utah CIHD as the new core. So I'll be co-directing this core with James Cox. So I'll tell you about the technique itself, the services that we provide, and then if we have time, I'll tell you about a novel vignette of biology that we're developing from it in central carbon metabolism. Next. All right, so the big picture here is really how the small molecule metabolome interacts with the proteome, right? So if we think about biology and boil it down to the fundamental domains of information, you have the genome, transcriptome, proteome, and metabolome. These don't exist in isolation. The information moves between these different domains and they interact with each other. Some of these arrows are very well characterized, you know, the central dogma of biology, you know, genome, transcriptome, proteome, etc. Other areas of interactions are much less characterized. And the one that I'm highlighting here is how small molecules interact with and functionally regulate or participate in protein function. And the reason why we just don't have very good knowledge of this interactome here is simply because there hasn't been you know, systematic platforms or technologies to really dive into this whereas the other ones are much better characterized. So you know, this interactome is relatively unexplored. You know, we hypothesize, and I think it's pretty, I think it's a relatively straightforward argument to make that small molecules and the metabolic environment of the cell, it would make sense that, you know, if there are acute changes in the nutritional status, so stress, you know, inflammation, etc., you could imagine that it'd be a very fast way to signal to the proteome via the small molecule milieu to regulate, you know, various cellular processes. And this is very well demonstrated throughout the literature, but it's usually on a protein per protein basis. And probably the best database or databases that we have capturing this information would be something like the KEGG metabolic database, where you're going from substrate to products via protein, or something like the Brenda database, which catalogs regulators, cofactors, etc. But again, I don't think the information is very well characterized, and we believe that the small molecule milieu controls a lot of proteome function. So I set out to develop a technology to detect these protein metabolite interactions with the hopes of, you know, gaining new insights on novel substrates, cofactors, products, and most importantly, regulators, looking for allosteric or orthosteric regulators. The big picture goal, this would be long-term, would be, you know, a systems level sort of understanding of how the metabolome and proteome interact. And from that, you could imagine that we'd have a much deeper understanding of how biology functions. But then on top of that, you could think of, if we knew of these interactions, you know, developing novel applications such as drug discovery and therapeutics to either directly target that protein or even modulate, you know, a specific pathway. So next, please. So here is the Midas platform. This is the nuts and bolts, and this would be the primary service that we would be providing through the protein metabolite discovery core, or Midas core. So the system itself is based on fundamentally a technique called equilibrium dialysis. So what we have, if we start up in the upper left, is a 96-well dialysis plate, and it has two sides to this plate. On the top side, you have the protein chamber, and to that you would add your highly purified, highly concentrated target protein or proteins. We then flip the plate over, and to the reverse side, we add defined pools of metabolite libraries. Our library is composed of 400 endogenous and exogenous human metabolites, extracted from the KEGG and Human Metabolic Database. So that's where we got all of our information for the metabolites. So it represents the core of human metabolism. The library itself is spread out in 4 pools, each roughly about 100 metabolites each. So we set this system up. Separating the protein chamber and the metabolite library chamber is a 10 molecular weight cutoff dialysis membrane. So essentially, this traps the protein in the top chamber, but the small molecule metabolites can freely diffuse across both chambers. We allow this system to reach equilibrium. If there is a direct interaction between a metabolite in our library and that target protein, what will happen, dependent on the KD and stoichiometry of that interaction, that small molecule will be relatively enriched. The concentration will increase in the protein chamber relative to the metabolite chamber. If there's no interaction, we'll have a rough equilibrium between both chambers. And then sometimes we also see depletions where a metabolite in the protein chamber appears to be removed or depleted in the context of the protein, but not in the metabolite chamber. So after equilibrium, we sample or separate the top protein chamber and the metabolite chamber. We crash out the protein on ice in 70% ice, 70% methanol, and then we sample the metabolite concentration in the top protein chamber and the metabolite chamber. And this is performed on a CIHD X500R ESI QTOF. And we're using this mass spec in a very high throughput manner. It's called flow injection. And essentially, we're not using any chromatography or column separation of the metabolites. It's a shotgun approach. So essentially, we pick up from the sample and then directly inject it into the source of the mass spec. And this allows all metabolites go through the source and hit the detector at the same time. And this allows for really rapid sample processing. So we're on the order of about 1 minute per sample. So that allows us to crank through many replicates across both chambers for many proteins in a relatively short order or period of time. From that, what it allows us to do is capture a relative concentration in the protein and metabolite chambers. And from that, we can calculate a fold change for that specific metabolite. And that's kind of schematized here in this intensity, mass charge versus intensity plot. So if we have a metabolite that's directly interacting with that protein, we'll see a relative enrichment viewed as the magenta line compared to the metabolite chamber. No interaction, they're the same. If there's a depletion, then we'll see that metabolite removed. And depletions can be viewed in a couple different ways. Substrate to product conversion. So if you provide an enzyme and that enzyme is active within our dialysis, it's possible, depending on the diffusion rate of the metabolite, that we'll actually see substrate being depleted and product being enriched. So that's one possibility of a negative fold change. Others include covalent modifications of that metabolite onto the target protein. So it's essentially pulled out or chemically modified and removed during the protein crash. A third option would be a very high affinity interaction that's not liberated during the methanol crash. We're assuming that the protein is being denatured and removed, but if that domain, which is binding that metabolite, is not disrupted, you could theoretically pull out the metabolite like a pull down assay, essentially. So we calculate the fold change across all 400 metabolites for your target protein. We pass them through our analytical pipeline that does 0.1% outlier removal, a PCA correction across the entire data set, and then it does stats. And how we define the statistical analysis is essentially your target protein versus our database of 220 plus proteins that we've screened. And from that database, we generate an average model. So we're assuming that not every protein is going to be interacting with every metabolite in our library. So that gives us a no signal control to then we can ask, okay, what is significantly enriched or positively, a positive fold change, or significantly depleted for your target protein. And from that, you will get this volcano plot, and this would be one of the forms of data that I would provide, where it tells you which metabolites are significant interactors with your target protein. I'll also add here, and this is what James briefly mentioned, another service, and this kind of gets towards the covalent modification aspect, where branching off up of the top right up there, you'll see NECM proteomics. So this would be non-enzymatic covalent modifications of the metabolites onto the target protein. And James and I have been piloting this approach to great success, where essentially we take the crashed out protein, because I'm not using that in the traditional MIDAS platform. I provide that to James, and then he can do essentially shotgun proteomics on it to identify specific PTMs or non-enzymatic covalent modifications to potentially reveal novel sites of interactions between the metabolite and protein. Next. These are some of the advantages. We don't need anything specially done to the protein. We can do it with or without affinity tags, etc. Same with metabolites. We don't need isotopically labeled metabolites. These are all just off the shelf from Sigma, Cayman, Avanti, etc. We can do, broadly speaking, both moderately hydrophobic and hydrophilic metabolites. We have a very high dynamic range. That's one of the, I would say, the big pluses to our technique relative to others, is that since we're based on dialysis, we can theoretically detect millimolar KD interactions and stronger. So anything stronger than that, we can definitely tell you. And this would make sense, especially for some metabolites in the cell. For example, ATP, is around 1 to 2 millimolar normally in the cell. So if you were to imagine a regulatory interaction between ATP and a target protein, well, you would imagine that KD would be around 1 millimolar. So our platform allows really low affinity detection. It's independent of protein functional assays. So, if a protein has multiple different cellular functions, so maybe it's interacts with other proteins, or it's an enzyme, etc., we can just tell you what metabolites we see interacting with your protein, regardless of what specific function it might be perturbing. It's multiplexed, so we're screening many metabolites at a time, not one protein per metabolite. It's large and scalable, flexible metabolite library. So if you have specific metabolites that are not in our library, we can theoretically include them in the screen. And then it's also amenable for high throughput analysis. And that was part of the bandwidth aspect. So, I say target protein, but we could easily screen through an entire pathway of proteins, in one shot in, a weekend with relative ease. We would just need the proteins. Next, please. And I think I'll just have to end here because I'm out of time. This is just nice validation of the platform. I'll have to save my novel biology for another time. But we worked with David Sabatini and Navitor from MIT and Boston, respectively. And we acquired Sestrin-2, Castor, and then Reb. So these are proteins that regulate the mTORC1 signaling pathway, and they integrate amino acid or nucleotide metabolisms into this pathway. And we screened all 3 of these proteins, and I think they serve a great validation of the platform. For Castor, which is known to bind and be inhibited by arginine. Arginine was our top hit and the most enriched metabolite. Sestrin-2 likewise enriched leucine and the leucine isoform family. So it'd be leucine, isoleucine, and allo isoleucine. And then also methionine is known to be an interactor, and we detected that as well. And then lastly, Reb is a GTPase, and we detected both GTP and so the substrate and product for Reb. So I think that's just a great demonstration that we can detect both, known substrate product enzymes interactions, but also novel allostery as well. So I think I'm out of time here. Maybe we could just zip to the acknowledgement slide. Cool. Before I thank everybody, I'll say that we are updating the protein metabolite core website. So I will have all of my information as far as what we need for samples, my contact information, protocols, etc., and pricing. All of that will be up shortly. So that's pending. So keep an eye on the Utah CIHD website. I need to thank all these people. So Jared, my mentor, who really was instrumental for developing this platform. My technicians, Paige and Sam. Ahmed, I didn't have time to tell you about the biology today, but we've done some really novel and cool, found some cool things in central carbon metabolism around lactate dehydrogenase. All of my collaborators that helped with protein production, conceptual, aspects, the Utah cores. So James Cox, metabolomics, proteomics core, the mutational generation detection core, and BioLo and the drug discovery core. I got to thank David and Navitor. And then literally the 220 plus proteins received was primarily from outside academic and commercial collaborators. So I'm indebted to them because they provided the protein, we provided them data, and we have numerous very productive collaborations spinning off of this platform. So that's been just a great interaction. The whole Rudder Lab with notable individuals there. And then, I need to thank the Utah Center for the Utah CIHD for funding this core. And then also HHMI, Calico Life Sciences, and Pfizer for also some aspects of funding this project. So thank you. And if you have any questions, I'd be happy to answer them.
Great. Thanks, Kevin. Appreciate that. Are there questions? Please unmute yourself and go ahead if you have them. So I have one. Have you done a membrane protein yet?
So we have not done a membrane protein yet, but I've got, the short answer is we have done membrane protein domains, right? So we've screened a number of those where, you might have a soluble cytoplasmic domain that's, fused to a membrane, integral membrane sequence. And usually how we approach those is either A, isolate that domain, right, and screen that, or fuse that domain to a solubility tag, such as, maltose binding protein. We've done a lot of those. Thioredoxin, that's another option as well, to basically increase solubility. Now, membrane proteins itself, Diane, this is the holy grail, and this is where we're headed. So I didn't have time to talk about it today, but we have a 3-year collaborative contract with Calico Life Sciences in the Bay Area. And our goal for the next 3 years is to take the Midas platform, expand the metabolite library, almost double it, and then adapt it so that we can put integral membrane proteins into the dialysis system. And that'll be either through, a detergent micelle sort of solubilization of the target protein, or probably preferably nanodisc. So essentially have your membrane protein in a nanodisc, put it into Midas, and then screen it. And this is going to require a lot of mass spec development. So we're actually adapting the current flow injection platform to a more conventional metabolomics. And then that'll allow us to kind of get through some of the biochemical caveats that come with working with integral membrane proteins. So hopefully, ask me again I don't know, we'll see where we're at in a couple months. Just keep hounding me, and then hopefully we can get your protein and other integral membrane proteins into the system.
Sounds good. Thank you. Anybody else? Feel free to unmute.
Are you able to get some sort of computational assistance in narrowing a range of metabolites that could bind to, say, a very well characterized protein? Meaning, if you have the portion of the protein that's well characterized, where you have all the atoms in there, the size, distance, electronegativities, could you have a computer assist in looking for parts of compounds that would fit or bind to it?
Yeah, so Hector, that's a great question. So I think you'd be referring to, in silico docking. So essentially taking the Midas hits, right, and then feeding them into a computer to see if there's a good docking site somewhere on your protein. That's not something that I do, but it's something that we could theoretically collaborate with. So we work pretty closely with Chris Hill's group here at University of Utah, and they've made us a lot of proteins. We've done various projects with them, and that's something that they could theoretically pursue. I mean, there's big caveats to that too, though, because docking it has both false positives and negatives associated with it. So I think you'd be kind of leveraging the two different platforms to hopefully point you in one direction. And we just haven't really fully completed that sort of analysis.
Thank you.
Great. We've gone a little bit over time. Does anybody else have a burning question they want to ask Kevin? Otherwise, I want to thank everybody for their participation and thanks for the presentations. And please feel free to reach out to these cores if you have questions. And thanks again for your time, everybody. Sorry for the presentation issues.
Thanks, Diane.
All right. See you later.